All right, these are your macromolecule notes, so let's go ahead and get started. All right, so of all the elements that make up living things, the most important are carbon, hydrogen, oxygen, and nitrogen, so CHON. And we're going to refer to CHON over and over again. It's just an easy kind of way to remember those four elements. We will talk about phosphorus and sulfur. Those are present in some parts of you, but in smaller numbers. So the main four are CHON, and kind of the top six are going to include phosphorus and sulfur. So as a whole, you can think of this as CHONPS, kind of a little word to help you remember it. Other atoms that play important roles in your body and in cell signaling and other stuff include calcium, sodium, potassium. You might recognize these as body salts. And we'll get to these as we go throughout the year, but for right now, I really do want you guys to just focus on carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. All right, so I do want to differentiate between organic and inorganic materials. So obviously we need to study carbon because all life on Earth does depend on carbon. We're carbon-based life forms. The other element that kind of science fiction has picked up on that might be useful for building bodies is silicon, which is why sometimes science fiction stories have silicon life forms, but at least for right now, nothing is as good as carbon is at forming bonds and building bodies. Even silicon doesn't quite come close to what carbon can do. Don't worry about the whole shell building too much with the electrons. All you need to know from this is that because carbon does need 8 electrons in its outer shell to form bonds with other atoms, it does this very easily, and carbon can actually bond to 4 other atoms. It can bond to an atom on the top, up here, it can bond to an atom kind of at the bottom, on the right, and on the left. And because it can bond with 4 other atoms, it makes these very, very large molecules very easily. All right, so we're going to differentiate between organic and inorganic compounds. So if something is organic or inorganic, it doesn't necessarily mean it has or doesn't have carbon. It does mean, if it's organic, that it has carbon-carbon and carbon-hydrogen bonds. It's not just good enough to have carbon, you must have those carbon-carbon, carbon-hydrogen bonds, right? So some examples, if we were together, I would ask you to give me some. But you've got lipids, carbs, proteins, DNA and RNA are examples of nucleic acid. We'll talk about those. But there are other molecules that might not be part of this list. Let's say I'm just going to kind of make up a molecule here. Right, as long as you have carbon-carbon, carbon-hydrogen bonds, you are organic. So in chemistry, I'm kind of making this up on the fly, but this is probably some kind of gas. This is not a carb per se, it's not a lipid or a protein or any of these other ones over here on the left. But this molecule that we kind of made up out of kind of thin air here, this would still be considered organic because it has those carbon-carbon and carbon-hydrogen bonds in it. All right. Inorganic compounds are not going to contain these carbon-carbon, carbon-hydrogen bonds. So for example, water obviously doesn't have any of those just simply because it has no carbon. Salt, table salt, right? Oxygen gas, even things like carbon dioxide, right? Even though carbon dioxide has a carbon in it, there are no carbon-carbon bonds or carbon-hydrogen bonds. There is only carbon-oxygen bonds. So even CO2 is considered to be inorganic. right? Nitrogen gas, which is most of the gas in the atmosphere that you breathe, right? Hydrogen gas, helium, that makes your voice sound kind of funny. At the bottom, it does say do not confuse with organic food. Food grown, it says, without pesticides, hormones, or fertilizers is considered organic. So this, the context matters here. Organic in the context of food means without pesticides, hormones, or fertilizers. It does not refer to the same kind of organic like we would talk about up here, organic versus inorganic compounds. So not talking about the same thing. Not talking about the same thing. All right. So there are four main building blocks of living things that are organic. So we call these things macromolecules. Macro means big, right? And molecule just means molecule. So these are large molecules in living cells. And we're going to think of these like giant kind of Lego buildings. There are four main types. And just like a major Lego building, you can break down these mega structures into individual Legos, right? So we're going to refer to these individual pieces, these kind of building blocks, if you will. We're going to refer to those as monomers, right? And the prefix mono means one. So if you link a bunch of monomers together, you will eventually get a big macromolecule. We call the macromolecules sometimes polymers. Poly just means many. Right, so if you link many, many monomers together, you'll get your polymer, your macromolecule. And those two words are basically just synonyms. Also, feel free to pause whenever you need to. That's the good thing about recordings. All right, here are your four macromolecules. So, a little pop culture reference over there. Your carbs, your lipids, your proteins, and your nucleic acids. So let's go ahead and talk about each of these in turn. Let's start with the carbs. So your carbohydrates, again, these are organic macromolecules, are mostly going to be your sugars and starches. We'll go over other examples too. But they are made of CHO, carbon, hydrogen, and oxygen. They don't have anything else in them. There's no nitrogen, no phosphorus, no sulfur. It's just CHO. And in a ratio of 1 to 2 to 1. Right? So for every one carbon, you're going to have 2 hydrogens and 1 oxygen. That ratio is very important. It's unique to carbs, right? The building block, so the individual Lego pieces of these giant monstrosities, is called a monosaccharide. Again, one, mono means one, and actually saccharide means sugar. So one sugar molecule, right? So there are 3 main monosaccharides: glucose, fructose, and this should say galactose over here. I think I've actually shown you glucose on the right over here twice. Glucose is actually the one that your body needs the most of because your mitochondria really like to use this to make ATP, which is energy. Fructose is the sugar that's found in fruit. Galactose is a monosaccharide sugar found in milk. And they do taste sweet. They fit in your taste buds. The receptors on your tongue fit these monosaccharides quite well. That's what they're adapted to detect, and this is why when you eat simple sugars like these monosaccharides, they taste sweet. So if you'll look over here to the right, sometimes you'll see these monosaccharides as a ring structure, like I'm showing you with this glucose here. Other times you'll see them as a straight chain structure, like with this glucose here. But if you count all of these atoms in both of these two structures, you will get 6 carbons, 12 hydrogens, and 6 oxygens. In both cases, these are C6H12O6. So if I didn't have these names, if let's say I crossed out these names and I asked you to tell me what these molecules were, and you counted the atoms, you would say, hey, there's 6 carbons, 12 hydrogens, and 6 oxygens. That's a 1 to 2 to 1 ratio. This must be a carbohydrate, and you would be right. What you're going to notice is that the other monosaccharides, fructose and galactose, are also C6H12O6. You're like, well, hold on, I thought C6H12O6 was only for glucose. And that's what you learned in middle school, but we're kind of expanding your awareness here. So any monosaccharide, the glucose, the fructose, or the galactose, actually all have C6H12O6. If you look at the visual, what you're going to realize is they're different monosaccharides, not because of the atoms, but because of where they are positioned, right? They just kind of structurally look different, right? Here you have a group that doesn't show up on your fructose. That's what makes the monosaccharides different, not the fact that they have different atoms. And we actually call this phenomenon, we call these to be isomers. If two molecules have the same atoms, but the only difference is their arrangement, those molecules are called isomers. If you link a bunch of monosaccharides together, you will get polysaccharides, your big structural carbohydrates. Again, poly means many. And there are three main types of polysaccharides: starch, cellulose, and glycogen, and we'll cover all of those next. I know this is kind of cut off, my apologies, but this says polysaccharides do not taste sweet. So I'll say they're not sweet, and that's because they are too big to fit on the receptors in your tongue. So here's how you get starch, right? All you have to do to get starch is link all of these glucoses together. So all of these ring structures, every single ring structure here is a glucose. So there's 1, 2, 3, 4, 5, 6, 7 plus glucoses, right? Starch is actually usually hundreds and hundreds, potentially thousands of glucoses linked together, right? And this is used for plant storage, right? This is how plants store the glucose. They link the glucose up and they store it. A really good example of this would be a potato, right? It's a big starch pouch that a plant uses to store that extra sugar. Another example of a polysaccharide is cellulose. This is also for plants. This is for structure. This is for structure. So the plant cell wall, I'll say plant cell wall is the main structure that uses cellulose. And if you look at the image of it, these are all glucoses. It's not a different polysaccharide because its building blocks are different. It's a different polysaccharide because of how they're linked together. Right here in starch, they're kind of holding hands towards the bottom. Here, the glucoses are kind of holding hands, if you will, kind of facing up. And they have these hydrogen bonds between different rows. So here you've got like a straight chain almost for starch. And then down here with your cellulose, you have like this very kind of lattice work of glucoses, right? So it's all glucose, it's just the difference is how it's organized, right? And at the bottom it says the cell wall of plants, of plant cells is made of cellulose. That word down here in the bottom is cellulose. All right, the last major polysaccharide that we're going to talk about is glycogen. Glycogen is animal storage. So plants stored extra glucose in the form of starch. Animals store extra glucose in the form of glycogen. And we keep this glycogen in one of two places. We keep it in our liver. Oops, I don't know why I did that, but whatever. And we keep it in our muscle cells. That is where we store most of our glycogen. And if you look at glycogen, each of these little hexagons here is glucose. So this is all still chains of glucose. The thing that makes it different is how the glucoses are connected. And we didn't really talk about what starch looks like, but I'll say it's more branched. It's more branched than starch. So here, if you're kind of looking at all, again, each of these little balls is supposed to be a glucose. See how this entire glycogen molecule has all of these branches? There's a branch, there's a branch. It looks kind of like a tree. There's a branch, there's a branch, there's a branch, right? Starch is similarly branched, but there are just less branches. So that's the main difference between those two. Again, glycogen for animals and starch for plants. Overall, carbs are usually a quick source of energy, right? If you guys are on sports teams, you might have like pasta dinners before a game. That's why. The pasta is a carbohydrate, and that's a great quick source of energy for your body right before you play whatever sport it is. Oops. All right, our second macromolecule that we're going to cover are lipids. Lipids are also made of carbon, hydrogen, and oxygen. The difference is that the ratio of the atoms is not 1 to 2 to 1. You've got a pretty significant number of carbons. You have a lot, so I'm going to do a double arrow. That's what I'm trying to show you here. The double arrow means a lot. A lot of hydrogens and minimal oxygen, so a down arrow to show that there's not a lot of oxygen, right? And again, these do tend to not want to mix with water because they are not polar, and we'll talk more about that too. But the basic structure of a lot of your lipids is this kind of glycerol fatty acid. So there's really kind of two monomers. You've got this glycerol molecule over here on the left, which has 3 carbons, 3 oxygens in it, 1, 2, 3, 4, 5, and 5 hydrogens. You don't have to remember that by any means. I'm just kind of showing you. That's not a 1 to 2 to 1 ratio. It's kind of close, but it's not quite. And then you've got these fatty acids, right? And if you count these, we don't have to memorize this at all because not even I have this stuff memorized, and these can all be different lengths, but 1, 2, 3, 4, 5, 6 carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, at least 10 hydrogens. We got, what do we got here? We got 1, 2 oxygens. Well, this oxygen's on it. There's 3 fatty acids here. There's one at the top, kind of one in the middle, and then you can't see it, but there is also one down there. So I think there's only just 1 oxygen. That's it. And if you're looking at this ratio, you're like, well, C6H10O1, that's CHO, but it's not a 1 to 2 to 1 ratio. So you would, even if you didn't know this was a fatty acid and you just counted the atoms, you should still be able to figure out that this must therefore be a lipid. It has to be a lipid because the ratio is way, way, way off, right? But anyway, so these are the monomers, the building blocks of lipids. You have your glycerols and your fatty acids. And notice the fatty acids are just a lot of carbons and hydrogens connected. These are definitely organic. Right? All right, so what do lipids do? They store long-term energy. They're good insulators. If you have animals that are adapted to the cold, they usually have kind of thick layers of fat or blubber. And that's all lipids. That's excellent insulators. The majority of your cell membranes are made of what's called a phospholipid, and we'll talk more about the cell membrane later on. They do have waterproofing qualities. Some steroids are lipids. Testosterone and estrogen are actually steroids, and they're lipids. Waxes, oils, vegetable, canola, olive oils, those are plant oils. Animal fats, lard, those are all lipids, right? Those are all lipids. All right, proteins. So proteins are made of carbon, hydrogen, oxygen, and nitrogen. So we're going to add on nitrogen. Some, not all of them, have sulfur. Again, sulfur is just the letter S. Okay. The building blocks of proteins, so the monomers of proteins, are called amino acids. And there are about 20 different amino acids. Depending on how they're all linked together, you can have different proteins. So in this little picture, each of these little pink spheres or circles is supposed to be, let me go back into that, is supposed to be an amino acid. Where'd my cursor go? There it goes. Right, so gly, ili, val, cis, glu, ala, these are all amino acids. And they are linked together. The links aren't shown, but there are covalent bonds between all of these amino acids called peptide bonds. So you can't really see them, but they are there. They are joined together by these covalent bonds called peptide bonds. Proteins, by the way, since we're talking about the word peptide, are also called polypeptides. So quite literally, many peptides is a synonym for protein. Don't memorize this at all. If you just were curious and you wanted to know what those amino acids looked like down to the atoms, you can go ahead and check this out, but we're going to skip that for right now. Let's do the function really quick. So proteins do function as building blocks of the body. Most of the material that builds bodies is made of protein. So for example, muscles are made of a protein called myoglobin. Hair and nails are made of a protein called keratin. In order to move, that your muscles need protein filaments called actin and myosin. There are proteins everywhere in your body, right? Some will regulate chemical reactions. Those are called enzymes, which we'll talk about later. Some regulate kind of how the body works as a whole, system-wide. So again, we talked about hormones a second ago. Some hormones, like testosterone and estrogen, are lipids, but others are actually proteins. So an example of a protein hormone is insulin, right? This is the same protein hormone that's responsible for regulating blood glucose levels. And if you have diabetes or know someone who does, chances are there's an issue with insulin, right? And the last function here is that they do function with defense and antibodies. So if you ever do have an infection, and you have antibodies that get sent out and attached to whatever foreign invader your body thinks is there. Those antibodies are all proteins. I know this is kind of cut off. This is immunoglobulin. You don't have to know that one. It's just an example. But you should know the basic functions of proteins. They're building blocks. They regulate chemical reactions. They regulate how the body works, and they serve as a defense mechanism. All right, the last major macromolecule are the nucleic acids. They are made of CHO, also nitrogen, and also phosphorus, right? They do not contain sulfur, so I will say no sulfur. That's a difference between them and proteins, right? Their building blocks are called nucleotides. And the nucleotide has three parts to it. It has this phosphate group, it has a 5-carbon sugar, and it has a nitrogenous base, right? This is DNA and RNA. We'll talk more about this later, but the macromolecule examples here, you either have DNA or RNA, right? So when you link all of these nucleotides together, you are building a DNA or an RNA molecule, and that's what this says down here. I know it's cut off. I'll fix that for next video, but that says DNA, and that says RNA. All right, the function of nucleic acids is that they store and transmit genetic information, right? They have genes, or what we would call genes, and therefore they control what happens in the body, they control what proteins are synthesized, etc., based on how the cell accesses that DNA or that RNA. And we'll have a whole unit on this later. This is a cute little trick. I don't know, everything has CHO. The proteins have the nitrogen. So do the nucleic acids, but because I guess there are two words here, they also have an extra letter. I don't know, how do you want to memorize that? Sulfur is not on here, so proteins also have sulfur. Maybe this trick works or maybe it doesn't, but you do need to know what atoms are in which macromolecules. All right, how do you attach monomers together to form these larger macromolecules? This process is called dehydration synthesis, and another name for this is condensation. So those are synonyms. Basically, we're going to remove a water. We're going to remove a water, because that's what dehydrate means, to synthesize, to put together. Right? So to make any of these, to make carbs, lipids, proteins, what we have to do is remove water in order to link these monomers together. Right? So we're going to get one monomer, we're going to have another monomer, we're going to release water, and we're going to make our large macromolecule. Right? This is called dehydration synthesis. This is how you build macromolecules out of their smaller monomers. So here's an example, right? Glucose is a monomer. It's a monosaccharide. It's the building block for a carbohydrate. If you take two of them and you remove a water, you can combine them to make malt sugar, maltose. That's what maltose looks like. If you have two sugars, like in this case, this is actually called a disaccharide. Di means two, and saccharide means sugar. Two sugars. 1, 2. Here's another example. So here we have two amino acids. So here's an amino acid on the left. Here's an amino acid on the right. Right now they're not connected. But if we take this OH and H, this oxygen and these two hydrogens, and we remove them together to make a water molecule, what happens is that this carbon and this nitrogen form a peptide bond, right? So that bond is right here. That's the new bond that we made. So let's do it one more time really quick. You've got two amino acids, 1, 2, and you know these are amino acids because they have nitrogen, right? They can't be lipids or carbs because they have nitrogen, which means they must be an amino acid or a nucleotide. And they're not nucleotides because they don't have phosphorus. So they must be amino acids. So there's one here, there's a second one here. Here's the water molecule that's going to get removed in the middle. And when those atoms are missing, this carbon and this nitrogen are going to form a covalent peptide bond right there. Now, we've linked two amino acids together, and we are building a protein. We are building a protein. Here are some more examples. Again, we walked through this a second ago, putting two monosaccharides together. In this case, we have glucose and fructose. Here is the water molecule that's going to get removed. So we remove that water molecule, and then the two individual monosaccharides get linked together. Alright, so that's how you build macromolecules. How do you break them? How do you destroy them? Well, you just put the water back in. If you took it out to build them, you add it to destroy them. This is called hydrolysis. So with hydrolysis, you are taking hydro, which is the water, and you are lysing, which means to break or to kill. So you're going to break these molecules Sorry, that's my dog chewing on something. You're going to break these molecules using water, right? By the way, Lysol, the hand cleaner or the disinfectant, is called Lysol because it lyses bacteria. It breaks and destroys bacterial cells. Pretty cool. All right, anyway. So here it's the exact opposite reaction. Here you have a large molecule. You're adding water back into it to break apart the individual components to get your two smaller molecules. So actually, let's look at this example. Right, so here you have sucrose. Sucrose is a disaccharide, and there are two monomers that make it. Here's a monomer, and here's a monomer. And I know it looks kind of funky, but those are two monosaccharides. So we're going to add a water. So here's our H2O molecule. One of the hydrogens will eventually go to one of the molecules. In this case, it'll go to the glucose. And the O and the H will go to one of the other molecules, in this case, the fructose. And it just splits them apart. It says no PDA. And the water comes in here, splits them apart. The water also gets split in half, by the way, right? Here's one hydrogen on the left, and here's another hydrogen and the oxygen on the right. So the water gets split up, but so do the monomers, right? So if you want to break down large molecules, you add water to them, and it breaks apart that if it's a polysaccharide, or if it's a protein, or if it's a lipid, it'll break it down into its monomers, its building blocks. All right, at the very start of these notes, we did talk about isomers. So just to end on that note too, again, you will have instances where you have molecules with the same chemical structure. In this case, C6H12O6. The difference between these molecules, like fructose, galactose, and glucose, is how those atoms are organized. And that means that all of these examples, all of these monosaccharides here, are isomers of each other. All right, so I know this was a long set of notes, but here's kind of the take-home points. You need to be aware of what a macromolecule is, what monomers are, and you need to know each macromolecule's building blocks, right? You need to know that monosaccharides build carbohydrates. You need to know that amino acids build proteins, that nucleic acids build that your nucleotides are going to build your nucleic acids, and that your glycerol and fatty acid chains are going to build your lipids, right? That's the main stuff. You need to be able to identify them visually. Like if I showed you a picture, let's say I showed you a picture of this, you should be able to figure out that it's some kind of carbohydrate because it has a C6H12O6, a 1 to 2 to 1 ratio, right? If there was a nitrogen in here, you should be able to figure out, hey, that's probably a protein. If it also had phosphorus, let's say maybe it kind of looked like this. Maybe it had a kind of a little sugary thing here, a phosphate group, and this nitrogenous base. If it has 3 parts in it, you should say, hey, that's got to be a nucleic acid. That has to be a nucleotide. That has to be a nucleotide. So focus on the basics. Focus on the basics. Don't get too overwhelmed with all the individual atoms, and don't even get too overwhelmed or focus too much on the hydrolysis and the dehydration synthesis stuff. We'll work on all of that. I know there's quite a lot of material here. I really do just want you to focus on the basics. So check out that practice, right? The practice will kind of help you focus, that practice packet will help you focus your attention on some of the more important material. I would also take a look at the Unit 2 overview that I've posted on the Google Classroom website. That also kind of takes the major concepts and distills them down for you to review. So we will go over this in class. We will spend at least a week on this content. Keep your questions for me, and I will see you guys next time.